So after studying the TS of testis, it will help us to understand the spermatogenesis process. Okay. So as we know, testis consist of 250 compartments of testicular lobules. Each testicular lobules are made up of 2 to 3 coiled seminiferous tubules. When we take out the section of testis, we'll get round shaped circular structures which are none other than the section of seminiferous tubule. We'll focus on one seminiferous tubule which is similar in all the seminiferous tubules. Okay. So these are seminiferous tubules which is lined by a basement membrane, and they are made up of, so the inner lining of seminiferous tubules are made up of 2 types of cells. So one cell is bigger in size, so we label, in same time we label. So basement membrane, this is Sertoli cells. Okay, and next thing is, one they are lined by Sertoli cells, another they are lined by male germ cells, or it is also called as gonial cells. Those are called as spermatogonial cells, which are called as germ cells, the one which undergo the spermatogenesis process. Male germ cell, gonial cell, or we'll write spermatogonia or gonial, spermatogonial cell, or spermatogonia also we can write. Okay, so basically, so inner side or inner lining of the not testis, it is one seminiferous tubule, section of seminiferous tubule. It is lined by 2 kinds of cells. One is a nurse cells called as Sertoli cells, and another is these spermatogonial cells, that is male germ cells. Okay, so once there is a division, okay, when there is division, these germ cells undergo meiotic divisions to form sperms, and they form different layers of different kinds of cells. Okay, I'll label one by one. So when the spermatogonial cells will divide, no, they form so these are slightly have drawn bigger because of when you study spermatogenesis, no, you will get to know the spermatogonial cells undergo growth phase and they grow in their size. Okay, and these cells now are called as primary spermatocyte. Okay, which is bigger than the spermatogonial cell. Next, all these spermatocyte, that is primary spermatocyte, undergo first mitotic division. Okay, that is first mitotic division, and they form two cells, sequential cells, and these cells are, okay, secondary spermatocyte. Now the secondary spermatocyte undergo second mitotic division and they form and these cells are called spermatids. Okay, these are spermatids. Now the spermatids undergo differentiation. There is no division. Okay, just compare the diagram with the spermatogenesis video. You will get to know. Okay, so soon after spermatids are formed, they undergo differentiation and they form okay, all these are sperms, or it is also called as spermatozoa. Okay, and the central part, that is this, we call it as hollow space, empty space within the seminiferous tubule, that we call it as lumen. Not only these sections, these things are there. So can you see the spaces between the seminiferous tubule, that is intercellular space? Okay, this space is also called as interstitial space. Okay, these interstitial space, within this interstitial space there are some cells. Okay, there are some cells. These cells are called as Leydig cell or interstitial cell. Okay, these cells are responsible to produce one of the male sex hormone, that is androgens or testosterone. So this is the structure of TS of testis. Okay, basically we can see the sections of seminiferous tubules, round shaped seminiferous tubules, where outer layer is called as basement membrane, okay, or geminal epithelium sometimes we call. Inner side of the seminiferous tubules lined by two kinds of cell. One is a nurse cell, another is this is spermatogonial cells. Those are sex cells or germ cells where they are able to produce the sperms. Okay, next, what is the function of Sertoli? Sertoli cells nourishes the developing sperm. See, here you can see the developmental stages. Okay, these are the different developmental stages which in turn produces sperms and this nurse cells will give the nourishment for the developing sperms. So next, first spermatogonial cells, what it does, they undergo growth, they increase in their size, and they form a type of cell, we call it as primary spermatocyte. The primary spermatocyte undergo first mitotic division to form two. Okay, each primary spermatocyte produces two secondary spermatocyte. These are secondary spermatocyte. These secondary spermatocyte again undergo second mitotic division to form spermatids. Okay, now what happens, these spermatids, they differentiate into sperms. They never undergo division, only they undergo metamorphosis or differentiation to form sperms. These sperms are attached with the Sertoli cells only. Okay, they release by the process called as spermiation, means the sperms are attached to the Sertoli cells, right? They get detached and within the lumen they are secreted and they travel. We know the route for it. From the lumen of the seminiferous tubule, where it goes first, rete testis, vasa efferentia, and that temporarily gets stored in epididymis. When there is need of ejaculation or insemination, from the epididymis they travel from vas deferens then to the urethra and glans penis. Okay, along with this there are one more cell which is important cell which helps to secrete male sex hormone, that is androgen or testosterone. Those cells are called as Leydig cell or interstitial cell, the one which present in the intercellular or interstitial spaces between the seminiferous tubules. Okay, so this is what the TS of testis.